Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. But before I start the topic, please like, share, and subscribe the channel. In the last session, we dealt with remers of aldoses. In today's session, we will start with furfural derivatives. Furfural derivatives, monosaccharides, when treated with concentrated sulfuric acid, undergo dehydration with the removal of three molecules of water. Therefore, hexoses give hydroxymethyl furfural and pentoses give furfural derivatives. The furfural derivative can condense with phenolic compounds to give colored products. This, form the, this forms the basis of knowledge test. Coming to the next point, reduction from alcohols. When treated with reducing agents such as sodium amalgam, hydrogen can reduce sugars. Aldose yields one alcohol, whereas ketose forms two alcohol because of appearance of a new asymmetric carbon atom in this process. Here we can see that glucose is converted to d-sorbitol due to change in first carbon atom having attached aldehyde to OH group. Here we can see that D-fructose can be converted to D-sorbitol and D-mannitol. Thus, glucose forms sorbitol, mannitol is reduced to mannitol, fructose becomes sorbitol and mannitol, galactose becomes dulcitol, and ribose to ribitol. Sorbitol, mannitol, and dulcitol are used to identify bacterial colonies. Mannitol is used to reduce intracranial tension by forced diuresis. The osmotic effect of sorbitol and dulcitol produces changes in tissues when they accumulate in abnormal amounts. Example, cataract of lens. Coming to the next point, glycosides. Where the hemiacetal group, that is hydroxyl group of anomeric carbon, is a monosaccharide, is condensed with an alcohol or phenol group, it is called as glycoside. The non-carbohydrate group is called aglycon. The glycosides do not reduce Benedict's agent and therefore because the sugar group is masked. In this diagram, we can see that methyl alpha D glucopyranoside and methyl beta D glucopyranoside is due to the changes in the carbon 1. They may hydrolyze by boiling into dilute acid so that sugar is free and then reduce copper. Alpha glycosides are reduced to maltase and form yeast, while beta glycosides are hydrolyzed by emulsin and form almonds. Enzyme hydrolysis thus affords a method to distinguishing between the two forms. Here are certain examples of glycosides. Glucose plus fluoridine forms fluorescein. Its source is rose bark and it causes renal damage. Galactose and lactose plus digitogenin form digitonin and its source is leaves of foxglove and it is a cardiac stimulant. And glucose plus indoxyl form plant indican. Its source is leaves of indigofera and it is a stain. Coming to the next point, formation of esters. Hydroxyl groups of sugars can be esterified to form acetates, propionates, benzoates, phosphates, etc. Sugar phosphates are of great biological importance. Metabolism of sugars inside the body starts with phosphorylation. Glucose 6 phosphate and glucose 1 phosphate are important intermediaries of glucose metabolism. Here we can see that glucose 1 phosphate and glucose 6 phosphate are due to change in carbon atom of attaching phosphoryl group. Coming to the next point, amino sugars. Amino sugars may be substituted for hydroxyl group of sugars to give rise to amino sugars. Generally, the amino group is added to the second carbon atom of hexoses. Amino sugar will not show reducing property and they will not produce OSA zones. Glucosamine is seen in hyaluronic acid, heparin, and blood group substances. Galactosamine is present in chondroitin and cartilage, bone, and tendons. Manosamine is a constituent of glycoproteins. The amino group is the sugar may be further acylated to produce N-acylated sugars such as N-acetyl glucosamine, that is GLUNAC, or N-acetyl galactosamine or GalNAc. These are important constituents of glycoproteins, mucopolysaccharides, and cell membrane antigens. Coming to the next point, deoxy sugars. Oxygen of hydroxyl group may be removed to form deoxy sugars. Deoxy sugars will not reduce and will not form OSA zones. L-fucose is present in blood group antigens and many other glycoproteins. Fuel gel staining is specific for 2-deoxy sugars and DNA in tissues. This is based on the reaction of 2-deoxy sugars with Schiff's reagents. Here we can see the structure of deoxy sugars as L-fucose and L-rhamnose. In this diagram we can see that sugars of nucleic acid such as D-ribose and 2-deoxy-D-ribose. Coming to the next point, pentoses. They are sugars containing 5 carbon atoms. Ribose is a constituent of RNA. Ribose is also seen in coenzymes such as ATP and NAD. Deoxyribose is a DNA. Ribulose is an intermediate of HMP shunt pathway. Pentose sugars. Arabinose is present in cherries and is seen in glycoproteins of the body. The name arabinose is derived as it was originally isolated from gum arabic. Xylose is a proteoglycans. Xylulose is an intermediate of uronic acid pathway. Coming to the next, disaccharides. When two monosaccharides are combined together by glycosidic linkage, a disaccharide is formed. The important disaccharides are sucrose, maltose and isomaltose, and lactose. Coming to the first disaccharide, sucrose. It is the sweetening agent known as cane sugar. It is present in sugar cane and various fruits. Sucrose contains glucose and fructose. Sucrose is not a reducing sugar and will not form OSA zone. This is because the linkage involves first carbon of glucose and the second carbon of fructose, and therefore free reducing groups are not available. Here we can see structure of glucose, that is alpha D glucosyl beta D fructoside. When sucrose is hydrolyzed, the products having reducing action, a sugar solution which is originally non-reducing but becomes reducing after hydrolysis is identified as sucrose. Hydrolysis of sucrose, optical rotation being plus 66.5 degree, will produce one molecule of glucose, optical rotation being plus 52.5 degree, and one molecule of fructose, optical rotation being minus 92 degree. Therefore, the products will change the dextrorotation to triborotation, or the plane of rotation is inverted. Equimolar mixture of glucose and fructose thus formed is called inverse sugar. The enzyme producing hydrolysis of sucrose is called sucrase or invertase. Honey contains invert sugar. Invert sugar is sweeter than sucrose. Coming to the second disaccharide, it is lactose. It is present in sugar. It is sugar present in milk. It is a reducing disaccharide. On hydrolysis, lactose yield galactose and glucose. Beta glycosidic linkage is present in lactose. The monomeric carbon atom of beta D glucose is attached to fourth hydroxyl group of glucose through beta 1,4 glycosidic linkage. The lactose may be alpha or beta variety depending on the configuration of first carbon of glucose moiety. Lactose form osazones which resembles badminton ball or hedgehog shaped or flower of touch-me-not plant. Lactose and lactate should not be confused. Lactose is the milk sugar, a disaccharide made up of galactose and glucose, whereas lactate and lactic acid are produced is a product of anaerobic metabolism of glucose. Coming to the third disaccharide, maltose. Maltose contains two glucose residues. There is alpha 1,4 linkage, that is, the anomeric first carbon atom of glucose is combined with fourth hydroxyl group of glucose through alpha glycosidic linkage. Here we can see the structure of maltose, such as glucosyl alpha 1,4 glucose. Maltose may be present, may be alpha or beta depending upon the configuration of first anomeric carbon atom. It is a reducing disaccharide. It forms petal-shaped crystals of maltosazone. Coming to the last of disaccharides, isomaltose. It is also a reducing sugar. It contains two glucose units combined with alpha 1,6 linkage. Thus, first carbon of one glucose residue is attached to sixth carbon atom of glucose through alpha glycosidic linkage. Partial hydrolysis of glycogen and starch produce isomaltose. Enzyme oligo 1,6 glucosidase is present in intestinal juice, can hydrolyze isomaltose into glucose units. In this diagram, we can see the structure of isomaltose, that is, two glucose residues in alpha 1,6 linkage. Here ends our session. Kindly like, share and subscribe the channel. Thank you.